All right, guys and gals, let's go ahead and pick up our discussion on the specific or the adaptive immune system. This is our third line of chemical defense within our body. And our adaptive immune system or specific immune system is going to help us to fight off specific species of bacteria or fungi or viruses. We remove specific microorganisms from our body with our specific or adaptive immune system. We will only use our adaptive immune system if the first and second barriers within the non-specific immune system have broken down. So let's look at this third line of defense. It's going to help us against both or any kind of non-specific pathogen. It helps protect us against cancer types, and it's going to depend on both the T and B lymphocytes that mature in the bone marrow or the thymus, particularly the red bone marrow, I should mention. So let's look at the B cells or the B lymphocytes. These B lymphocytes, when activated by the specific pathogen they are designed to fight off, are going to start reproducing and dividing many times over. And as this B lymphocyte is reproducing, it turns into a plasma cell and a memory cell. Plasma cells you can think of as an antibody factory. A plasma cell just pumps out tens of thousands of antibodies per hour designed to bind to a specific microorganism and eliminate that microorganism. And then, well, I shouldn't say necessarily eliminate, but neutralize that microorganism. And then we have memory cells. Memory cells are what give us immune memory. They help us to fight off an infection the next time we're exposed to that specific pathogen. Memory cells are what we want to make when we give somebody a vaccine. When we vaccinate somebody, we expose them to a weakened version or part of a pathogen that typically doesn't make them get sick, but does cause a B cell to differentiate to form the plasma and memory cells. We also have T cells. And there are over 200 different kinds of T cells that have been identified. Your textbook focuses on the three big, the memory, the cytotoxic, and the helper T cells. When you think of a cytotoxic, aka a killer T cell or cytotoxic T cell is going to kill a cell infected with a virus or will kill a cancerous cell. When we look at B lymphocytes will differentiate to plasma cells and make antibodies, and these antibodies function really well for destroying bacterial infections or neutralizing a bacterial infection. When we have a viral infection, we need to rely on the cytotoxic T cells to destroy the human cells infected by the virus. We also have a helper T cell. The helper T cell is going to be the coordinator of the immune system. It activates both B and T lymphocytes. And then we have the memory T cells. A memory T cell is sometimes called a T. We have both, sometimes going to be derived into several different subcategories. We'll leave it at that right now. This is an introductory course. And the memory T cell can allow for active response and reactivation of the immune system from a cytotoxic perspective. The memory T cells are very analogous to the memory cells or the memory B cells that are made from the B lymphocytes. They allow for that quick response when they were exposed to the specific pathogen in additional time. So if we look at our adaptive defenses, if we activate a B cell, that B cell needs to get activated by either having an antigen-presenting cell, which is typically going to be some kind of phagocytotically active cell, showing the T helper cell what is in the body, and that T helper cell can then activate the B cell directly, or the antigen floating around in the surface can activate the B cell directly. And the B cells will have a receptor protein on their surface known as a B cell receptor that recognizes the specific antigen of the specific pathogen. And then that B cell will differentiate and form memory B and plasma cells. Plasma cells pump out lots of antibodies into the body. And memory cells hang around and are able to be actively turned on to generate many plasma cells on the drop of a dime. If we look at a cell that's infected with a virus, that cell that's infected with a virus can activate a T cytotoxic cell, and that T cytotoxic cell will then destroy virally infected cells. And that T cytotoxic cell can also differentiate to a memory T cell as well. It's worth emphasizing that on the surface of a T lymphocyte, we have a T cell receptor protein. B cell receptors and T cell receptors are structurally very different from each other. So let's look at these B cells. B cells help us by making antibodies. And then the antibodies can bind to the pathogens to help neutralize, not destroy, but neutralize the pathogens. We make B cells within our red bone marrow, and the B cells mature within our red bone marrow. Our B cells are capable of directly recognizing an antigen, or they're capable of being activated by a T helper cell. Once activated, a B cell undergoes a process called clonal selection. What is clonal selection and clonal expansion? That's just a nice way of saying it makes lots of copies of itself. It reproduces over and over and over and over and over again. It doubles and doubles and doubles and doubles until we have tens of thousands of plasma and tens of hundreds of memory B cells floating around within the body. Each individual B cell has a unique receptor protein known as a B cell receptor. That B cell receptor can only bind to a specific antigen or a specific molecule that's associated with a specific pathogen. Once a B cell receptor binds to the specific antigen that activates it, this is going to cause the B cell to be activated. We also can have the helper T cell expose the B cell to the specific antigen to activate itself. Most of the B cells that are going to be produced via clonal selection and clonal reproduction are going to turn into plasma cells, those antibody factories. And then when the plasma cells have a short lifespan where they secrete many tens of thousands of antibodies per hour, and then the sum of those B cells will differentiate into memory cells, which have a lifespan of years to decades. So those memory cells give us long-term immunity. After an infection has passed, the plasma cells will destroy themselves. They pop. They go through apoptosis, and only those memory cells will remain. So here's a generic B cell. We have the specific antigen binding to the correct receptor. And if we look, we can see there's three different B cell receptors. We have the circular, the triangular, and the square B cell receptors. If we have the correct antigen bind, we'll have activation. And the B cell receptor for the circular antigen will receive that circular antigen, and we have clonal selection, so we have lots of and clonal expansion. And this particular B cell makes many copies of itself, and it turns into many plasma cells that pump out antibodies, and it will turn into many memory B cells. To help activate a B cell, a cytokine from a T helper cell will further enhance the activation. After the infection has passed, the plasma cells go through apoptosis. They have pre-programmed cell death and pop, leaving only those memory B cells. If we look at a typical or generic antibody, they're going to have repeating units of a Y-shaped protein. The trunk of this Y-shaped protein, and let's back up here. Here's the Y-shaped protein I'm talking about right here. So we have 1, 2, 3, 4 antibodies that I just highlighted. The trunk of that Y-shaped protein is the constant region. It's the same from antigen to antigen and antibody to antibody. Now, if we look at the ends of this Y-shaped protein, that's the variable region that binds to the specific antigen. This is where the specificity comes in for the specific or the adaptive immune system. So here's the constant region, the heavy chain, that always remains the same, and then we have the variable region at the end that can bind to the specific antigen. There are 5 different classes of antibodies: IgG, IgM, IgA, IgD, and IgE. When I use this abbreviation Ig, this is an abbreviation for the technical term for antibody. Antibody is not a technical term. The technical term is immunoglobulin. We have immunoglobulin G, immunoglobulin M, immunoglobulin A, immunoglobulin D, and immunoglobulin E, or Ig for short. If we look at IgG, immunoglobulin G, that's the most common and the smallest antibody. Immunoglobulin M is going to be a large antibody. It activates the complement system and is the first antibody formed with any new infection. IgA is an antibody that's primarily associated with secretions in the body, both breast milk and mucus secretions in the body. IgD is an antibody that's found on the surface of B cells. IgD is the B cell receptor protein. And then we have IgE. IgE is going to be found as an antigen receptor within mast cells and tissues. So both IgD and IgE are surface cell receptors. IgG is the common antibody, IgM is the first one on the scene, and then IgA is the antibody that's in bodily secretions. How do we make monoclonal antibodies, or many copies of an antibody? Well, first, we are going to need to get a B cell, or a plasma cell that's derived from a B cell. And once we isolate that plasma cell, we are going to need to take that plasma cell and cause it to live forever. Plasma cells don't live for a very long time. They have a pre-programmed cell death or apoptosis event that takes place. So we'll take that plasma cell and fuse it with a cancer cell, a myeloma cell. And by fusing that plasma cell with the myeloma cell, we create a mutant hybrid cell. And this mutant hybrid cell is called a hybridoma. What is this hybridoma? This hybridoma is going to be an immortal cell line of plasma cells that will pump out one specific kind of antibody. So we take our favorite plasma cell and we bind it with our favorite cancer cell to come up with the hybrid. And what does the hybrid do? The hybrid never dies. The hybrid continually reproduces and continually secretes our favorite antibody. What can we do with our favorite antibody? Well, we can have it be an antivenom for a snake bite, antivenom for a spider bite, or we can use it for passive immunotherapy. For example, negative blood type mothers are injected with the antibody that neutralizes positive blood types while those mothers are pregnant. This is called the ROGAM shot, and this is a standard procedure to help prevent mom from having her immune system exposed to the rhesus factor and potentially killing off second, third, or fourth, or fifth children. More on that miscarriage later. So what are some of the characteristics of T cells? If we look at a T cell or a T lymphocyte, it's going to be matured in the thymus. They are produced in the red bone marrow and maturate within the thymus. And recall, the thymus is directly around the base of the trachea, right where the primary bronchi leave left-right primary bronchi, and it's large in newborns and gets smaller and smaller and smaller in adults. To activate a T cell, we need to have the correct antigen presented within the groove of an HLA, aka MHC. HLA stands for human lymphatic associated molecule. MHC is for major histocompatibility complex. And actually, don't quote me on that HLA. I really don't use that term HLA very often. MHC is the one that I personally use all the time, major histocompatibility complex molecule. So when our favorite antigen is put in the correct MHC molecule of the correct T cell, the T cell is activated and it will start to reproduce. It will reproduce into cytotoxic T cells. What do cytotoxic T cells do? They destroy human cells that are infected with a pathogen. Another way of saying that is they destroy non-self antigen bearing cells, which is kind of a technical way of saying that if you have a human cell infected with a virus, we need to kill it with a cytotoxic T cell. We also have helper T cells. Helper T cells are going to secrete cytokines, and these are a different kind of cytokines compared to the ones that we mentioned earlier with the non-specific immune response. These cytokines from the helper T cells are going to help to activate other cells of the immune system. Each T cell is going to have a specific receptor known as a T cell receptor. This T cell receptor is going to bind to your favorite antigen with the help of an antigen-presenting cell, or APC. When we think of a typical APC or antigen-presenting cell, these are going to be the phagocytotically active cells from the non-specific immune system. And after the macrophage or the neutrophil or the dendritic cell binds to and swallows and phagocytizes the pathogen, it'll take bits and pieces of that pathogen, rip them apart, and express them on the surface of its cell membrane, and then carry them over to the T lymphocyte and present that antigen to the T lymphocyte, hence the term APC, antigen presenting cell. It'll take that antigen and put it into a major histocompatibility complex, also known as the human leukocyte antigen. So when I tried to tell you what HLA stood for on the previous slide, I totally got it wrong. It's a human leukocyte antigen, also known as a major histocompatibility complex. Most individuals I know use the term MHC. Very few people that I know of use the term HLA. Once our T cell binds to the specific MHC or HLA protein that activates it, it will go through clonal expansion. This causes there to be many helper T, many cytotoxic T, and a couple memory T cells produced. After our infection is passed, it's important that the cytotoxic T cells are shut down. And to help shut down the cytotoxic T cells, our helper T cells will induce apoptosis for those cytotoxic T cells, leaving the memory cells behind. So here's our favorite T cell, just a generic T cell, and the macrophage phagocytizes the bacterium and then takes a chunk of the bacterium and puts it on its surface within a recognizing MHC category 1 complex. We activate, where'd my mouse go? There it went. We activate the T cell. The T cell then reproduces and makes cytotoxic T cells and memory T cells. The cytotoxic T cells then go through apoptosis, and the memory T cells are left behind. Helper T cells are specifically the T cell type that secretes cytokines to help activate other cells of the immune system, both cytotoxic and B lymphocytes. And if we look at the cytotoxic T cells, they function very specifically by secreting both granulozymes and perforins. The granulozyme is a specific kind of enzyme that will initiate the chemical cascade to trigger apoptosis within a human cell. A perforin is going to be a pore that perforates a human cell. You could think of a perforin as the complement system for human cells infected by viruses. It causes the human cell to swell up and pop. So, guys, gals, that was a very brief, very quick overview of the adaptive immune system. Which of the following is incorrectly paired? We have plasma cells with antibody production, T helper cells with immune memory, T cytotoxic cells with destroying other cells, and antibody for targeting antigens for destruction. Go ahead and pause this video, rewind it, and get me an answer, or check your textbook. 5, 4, 3, 2, 1. The correct answer is T helper cell and immune memory. A T memory cell aids us with immune memory. T helper cells specifically activate T cytotoxic cells and B lymphocytes. Once the B lymphocyte is activated, it forms plasma cells. So that is all we have for the adaptive immune system. If you have any questions about the adaptive immune system, please feel free to post them in the class discussion board, shoot me an email, or swing by my office when you're on campus. Happy studies!